whole seminar series. So before, no, I will introduce our speaker today. Before he speaks, I will introduce him. It's my pleasure to introduce Duncan Menge for this week's seminar. So Duncan is a professor at Columbia, but almost newly hired. You probably still have closed boxes in your office and lab, so he's been there for a year. So Duncan, well, I still have boxes in my office.
Say at least one person I talked to today, I still had boxes in there.
So Duncan did his undergrad at Stanford, and he grew up on the West Coast. He did his undergrad at Stanford, and then he moved to Princeton for his PhD to work with Simon Levin and everyone there. So usually you work with more than one person, or in a small environment with Steve McCullough and Lars Hedden. I guess you're interacting from it with him. And then he moved for a postdoc to NCs, to the West Coast, was still on the West Coast. And went back to Princeton for a postdoc also, and until he took his position at Columbia. And so Duncan works today, so he has a real lab, a wet lab, and he has, he's doing theory also, and working on ecosystem theory, but from this very small scale, doing dynamical theories of ecosystems, to global cycles, biogeochemical cycles, working with more practical, more very concrete issues about nitrogen limitation, nitrogen cycles. I think your talk today will be more on the large scale end of the spectrum. I'll leave it to you. So.
Great. Thank you very much for it.
So there will be a wine and cheese if you want to talk to Duncan after, if I don't take all this time. There is a wine and cheese after the seminar.
Thanks, Fred. Thank everyone for coming. It's been a lot of fun to chat with some people today. I look forward to hopefully chatting with more people later. So as Fred mentioned, I work on ecosystem ecology, among other things. The main thrust of my research recently has been focusing on the role of charismatic meganutrients, nitrogen and phosphorus, in ecosystems. And I work on those largely because I find them intellectually fascinating. There are a lot of fun puzzles to try to figure out, and that's fun. I initially started working on ecosystem ecology from a global change perspective, working in a global change experiment in California. And nutrients are very important for global change type issues. This is a graph that most of you have probably seen some version of at some point. Atmospheric carbon dioxide is rising. If you haven't heard that, then please look it up. Google it. One of the things we really want to know about rising atmospheric CO2 is the extent to which this is going to lead to more plant growth. So this is something we call CO2 fertilization. Plants need CO2 to photosynthesize. If they have more CO2, they might grow more. If they do, if CO2 fertilization is happening, that means that there's less CO2 in the atmosphere, which means that we get some help with the climate problem. If they don't undergo CO2 fertilization, then we have less help with the climate problem. So this is something we really want to know. The now not the most recent IPCC report, but the 2007 one, said that the rate at which CO2 fertilization saturates in terrestrial systems dominates the present uncertainty in the role of biospheric feedback to rising atmospheric CO2. So from the perspective of global Earth system models, this is one of the things that we know the least well. So what controls CO2 fertilization? So what does control? Empirically, we know that there are a bunch of things that do. One of those things is nutrients. We know that nutrients are important. People can get away with charging money for horse poo, even if it is the best kind, semi-rotten. And they do that because horse poo has nutrients in it. If you put that on plants, they grow more. And if you're trying to sell your plants, they grow more. If you're trying to understand global biogeochemistry, you want to know whether plants are going to grow more and take more CO2 out of the atmosphere. So we need to know what's controlling CO2 fertilization. We know that nutrients can play some role. That's not new knowledge. What is tricky, though, is that nutrient cycles are very complicated. The nitrogen cycle, sort of like the Krebs cycle, is something that most intro bio students memorize because they have to for the test, and then they forget about it because there are too many things to remember. So trying to get a good enough understanding of that such that we can plug it into global models and make quantitatively accurate predictions about what's going to happen in the future is a big challenge. And that's something that, to me, says now is the perfect time to be working on ecosystem theory, trying to understand the fundamental questions in what controls, under what conditions nitrogen controls plant growth and by how much, and under what conditions it's phosphorus. Trying to get that theory to a point that we can plug it into the big Earth system models. So today I'm going to be talking to you about that fundamental theory aspect of it in the context of large-scale patterns in global biogeochemistry. I'm also doing some work with one of the groups that runs one of the big IPCC Earth system models. So I'm not talking about that today, but the work that I'm doing today is feeding into that. So we can talk more about that later if anyone wants. For the talk today, I'm going to have 4 sections. First, I'm going to show you some latitudinal patterns in global biogeochemistry. Patterns are fun, but like my 3-year-old daughter, I like to ask the question, why? So she asks why all the time, as many of you who had kids probably know. So I'll then ask, why do we see the patterns we do? Why do we see persistent nitrogen limitation, mostly at higher latitudes? Seems kind of weird for reasons I'll talk about. The flip side is that in tropical latitudes, we tend to see more nitrogen than we think should be there. So why is there nitrogen richness? Why is there more nitrogen there than we think there should be? And then I'll put it together and try to understand, at least make a first stab at trying to understand why we see this transition at high latitudes from nitrogen limitation, low latitudes to nitrogen richness. A lot of what I'm talking about today is going to revolve around the process of nitrogen fixation. So again, most of you probably learned this in intro bio, but I'm going to assume that you don't remember all of the details. This is a process that only prokaryotes do, so only bacteria can do this. They often do that in symbiosis with higher plants. So what I'm showing here is, so this is a root nodule here of a plant called Coriaria arborea. This is plant tissue on the outside, but bacterial cells in the inside. And in this relationship, the bacteria essentially act as nitrogen fixation factories. Plants send sugar down to the bacteria send nitrogen back up, and it's a nice happy symbiosis. Or at least it is in textbooks. Nitrogen gas is the substrate there, so 78% of what you're breathing right now is nitrogen gas. You do absolutely nothing with it. You breathe it in, you breathe it out. It does nothing to you or for you. These bacteria are the only ones that can use it. They take that nitrogen and turn it into a form of nitrogen that is useful for plants. They then send that nitrogen to the plant in return for carbon. There are 2 different types of plant bacteria relationships that I'll talk a little bit about today. One of them is actinorhizal, so this is plants that are not legumes, so they're not in the pea family. There are other types of plants that form symbioses with one type of bacteria called Frankia, so it's not a legume. And the other type is the one that most people are more familiar with. Rhizobial symbioses are legume plants forming symbiosis with another type of bacteria, which is called rhizobia generally. All right, that's what nitrogen fixation is. So let's talk about latitudinal patterns in global biogeochemistry. I'm going to show you 3 patterns. The first one is this. So this is looking in streams draining temperate forests and streams draining tropical forests. On the y-axis, I'm showing you the concentration of inorganic nitrogen. D is dissolved inorganic nitrogen in streams. And from a terrestrial perspective, we look at the nitrogen we see in streams and we interpret that as a loss from the forest. So these are all samples that have been taken right when the stream begins. And how we typically interpret this is if there's a lot of inorganic nitrogen in the streams, that means the plants are allowing a lot of nitrogen to leak past their roots. Plants are very good at taking what they need out of the soil, and if there's a lot of nitrogen leaking past the roots, that means that they don't need that nitrogen. So when we look at this figure, note that this is a log scale. We see that temperate forests, for the most part, have much lower nitrogen concentrations leaking out of the roots than tropical forests do. And we generally interpret this as temperate forests are much more nitrogen limited. So this is one piece of evidence. We have a bunch of other evidence for this conclusion, but generally we think that temperate forests are nitrogen limited. If we give them more nitrogen, they tend to grow more. And a lot of tropical forests, at least, are not. A lot of them leak a lot of nitrogen out of their, past their roots. So that's pattern number 1. Tropical forests seem to be more nitrogen rich. They have a lot more nitrogen available. The next pattern has to do with the abundance of nitrogen fixing trees. So to set this one up a little bit, nitrogen fixation is a process that is energetically expensive. To break that triple bond of the nitrogen molecule takes a lot of energy. It takes 16 ATP to break that. So we think that this process should only really make sense, should only really be selected for when nitrogen is very rare, when it's very scarce. Looking back to this, that says that we think nitrogen fixation should be very advantageous here in temperate forests, because that's where there's not a lot of nitrogen around, that's where these plants should have a competitive advantage. This figure shows the percent of the basal area, so these are data from forest inventories in the United States and Mexico, in the dots, and in a bunch of countries in Amazonia that are between these bounds. So this shows the percentage of basal area, which is, you can think about that as the fraction of the community biomass, that is comprised by nitrogen-fixing trees. And it's showing this as a function of latitude. So here's the equator, here's Amazonia. So Amazonia has approximately 10% of basal area being N-fixers. If you go into Mexico, it's on average about 10% until you hit about between 30 and 35 degrees latitude, and then N-fixers drop out. And there are almost no N-fixers at high latitudes. Now remember, high latitudes is where we think they should have an advantage, but that's exactly the opposite of what we see. They're very rare where nitrogen is very scarce, which is weird. The third pattern is related to that one. This is the successional abundance distribution of nitrogen-fixing trees at those different latitudes. So at higher latitudes, these are now coterminous US forest inventory data. This is looking at stand age on the x-axis for both the eastern US and the western US, percentage of basal area on the y-axis, and different types of N-fixers shown in different lines. Note that this one is not 100%, but that is 1%, meaning less than 1% of the basal area here is N-fixers. And the successional pattern, regardless of where we look in the US, and we think more broadly, is that N-fixers decrease through succession. Where they are present, they're only really present early in succession, but late in succession, they're rare pretty much everywhere. Despite the fact that those late successional forests are often nitrogen limited. So again, very weird pattern of no N-fixers where we think they should be. In the tropics, things are a little bit different. N-fixers overall are much more abundant, and these are some data looking at successional patterns. So all the figures I'm showing you here have something like stand age on the x-axis and something like biomass of N-fixers, or in this case, legumes on the y-axis. One common misconception, most people think that all legumes can fix nitrogen. That is false. Only about 2/3 of legume species can fix nitrogen. So here, this is legume biomass. In this paper, there is a table that showed the ones that can fix nitrogen. So it says that the proportion of the community that is N-fixers is about flat until there, and then it jumps up to about there if you're looking at just the ones that can fix nitrogen. So take home, N-fixers don't decrease during succession in these sites in, I believe this was in Brazil. These are sites from Panama. Again, same basic story, N-fixers do not decrease in abundance through succession. If anything, they might increase. And these are some data from Costa Rica showing the same basic thing. So these are N-fixer percent of basal area across stand age, and if anything, they increase through succession, even though we think that if nitrogen limitation is anywhere here, it's going to be early on, and these parts of the forest should not at all be nitrogen limited, but that's where the N-fixers are. So a bunch of weird latitudinal patterns. We have nitrogen being much more abundant at lower latitudes. We have N-fixers being more abundant at lower latitudes, even though we think they should be more abundant at higher latitudes. And they last through succession much better at lower latitudes. So if we want to look at the temperate side, we might want to figure out, before we go to why do we see this latitudinal transition, we first want to understand why could we see persistent N limitation and no N-fixers at all, and why could we see persistent N richness and N-fixers doing okay there at all. So I'm going to look at those next. So first, let's look at the N-limited case. So when we see N limitation, why might we not see any N-fixers, and why might they not do well late in succession? Some theoretical work I've done on this follows up on some ideas that Peter Batusik and Bob Howarth had a long time ago. They essentially said that understanding the persistence of N limitation boils down to what prevents N-fixers from invading the system? Why are they not there? And theoretical work I have done essentially confirms that, saying that's what we need to understand. So I'm not going to go into details of that, but if we want to understand the question, why does end limitation persist, we need to understand why have late successional end fixers not evolved. So why aren't there any end fixers in late successional temperate forests? There are a couple of different ways that we could address that. I'm going to show you a few of them. The first is using forest inventory data. I'm going to speed through this part. So the basic question here is there a phylogenetic constraint to there being late successional end fixers? Maybe it's just the case that there has never been a late successional tree that has both been late successional and had the ability to fix nitrogen. I have the slides after the talk, but I'm just going to give you the result here. The answer is no. There are plenty of trees that are in northern latitudes that are in the end-fixing clade that are late successional. So it does not seem to be the case that there are phylogenetic constraints to being a late successional end fixer. The next thing we might want to understand is, from a theoretical perspective, what might lead to selection against end fixation in an end-limited environment. So the problem is typically set up as being counterintuitive. We say that when nitrogen is limiting, end fixers should have an advantage. And this is trying to understand under what conditions might that not be the case. So I have used a model to try to get at this question. And I'm going to talk about a few different versions of a model in this talk, so I'll spend some time now to set up the basic structure of the model. We're looking at pools of nitrogen, or occasionally carbon and phosphorus as well, and fluxes. So the boxes are where the nitrogen is, the arrows are where it's going. We're considering the case where there's nitrogen in biomass, so this is plants, and we're not differentiating between different types of plants or different tissues. There are two forms of nitrogen that we're interested in the soil. One is available nitrogen, so the type that plants can get at. That's things like nitrate and ammonium. There's also unavailable nitrogen, so big organic molecules that the plants can't take up. That could mean actual chunks of leaf that are sitting there, or it could just mean the molecules themselves that are too big to be taken up. So this is a very toned down version of the nitrogen cycle. It's in the soil, plants can take it up. When plants drop their leaves, that goes back into the soil, and eventually that gets mineralized back to available nitrogen. There are also inputs and losses to and from this system. So there are some external inputs to the available pool. For nitrogen, this is mostly atmospheric deposition, so you can measure nitrogen that's coming in rain, and that always comes in at some small level. We can collect that with collectors. There are losses from both of the soil pools. There are always some leaching losses, so water goes through the soil, that leaks out past the roots and gets into streams. And finally, there's nitrogen fixation. So this is the one form of input that plants really have any control over, and they do that through these symbiotic relationships with bacteria. And this is only certain plants that can do this. So what we wanted to ask with this model for this particular project was, under what condition can nitrogen fixation not be selectively advantageous late in succession? So if we want to understand late succession, we're going to start with a late successional forest. So this is a 500-year-old forest in New Zealand where I did all my PhD research. And we take an evolutionarily stable strategy approach to this. So we ask the question, what strategy of fixing nitrogen, either not fixing nitrogen or fixing some, is evolutionarily stable under different contexts. So what that says is, consider a forest that has some strategy and then allow some mutant to appear, and that mutant has a different strategy. It's fixing more or less than whatever's there to begin with. We're looking at the conditions under which that mutant can invade or not, and if we can find a case where non-fixation cannot be invaded, then that means non-fixation is evolutionarily stable. So in the model, we built in trade-offs between end fixation and other things plants do. One of them that's easy to think about, and the one that I'll focus on here, is the trade-off between building nodules and paying bacteria to fix nitrogen versus building fine roots and paying either mycorrhizae or bacteria in the soil to help get nitrogen in another way. So plants don't have an infinite amount of resources. They have a finite amount of resources, and they can only, with one bit of resource, allocate toward fixing nitrogen or allocate towards taking up nitrogen from other sources. If you allow there to be these sorts of trade-offs in the model, then you can also ask the question, what happens when those trade-offs vary in strength? there could be strong trade-offs, meaning allocating a little bit toward fixing nitrogen means you have a strong decrease in the ability to get nitrogen from other parts of the soil. Or they could be weak, maybe fixing a little bit of nitrogen has little effect on how much nitrogen you can get from the soil. When trade-offs are weak, we get the situation that seems intuitive. So this says that this is the amount of nitrogen fixed by a strategy on the x-axis and the fitness of the mutant relative to the non-fixer on the y-axis. So what this says is that non-fixers have the same fitness as non-fixers, that's the 0, 0 point. And then as more fixation happens, the fitness goes up. So that says in n-limited scenarios, fixation makes sense. One thing that I think I haven't mentioned, that this particular model assumes that nitrogen is limiting. So it's setting up, and I assume nitrogen is limiting not because I think it always is, but because, number one, that's the situation we think is most common, and number two, if we can understand a situation where nitrogen fixation doesn't make sense in an n-limited system, then it's very easy to understand it in a non-n-limited system. So this is setting it up to get a conservative answer. When trade-offs are strong, though, so when fixing a little bit of nitrogen makes it very hard to take up a lot of nitrogen from the soil, then you can get a scenario where it doesn't make sense to fix nitrogen. So the more nitrogen is fixed, the less, or the lower fitness of the fixer is. And importantly, we can get quantitatively helpful predictions for this. So what we're trying to understand is a situation in which non-fixation is evolutionarily stable. And that can be looked at like this. So we can say for that little bit of nitrogen fixed, so going from a non-fixer to a fixer that's fixing just a little bit, what is the change in biomass from fixing that nitrogen relative to the percent change in nitrogen uptake, which is then translated through to a change in biomass itself from that new nitrogen they get. So if the percent increase in biomass from that nitrogen fixed is less than the percent decrease in nitrogen uptake, then that means non-fixation is evolutionarily stable. And we can try to put some numbers on that, so we looked in the literature. And our best guesses are that 1 unit of nitrogen fixed leads to about a 10% increase in biomass, so that helps. But the structural costs of fixing nitrogen, so this means building nodules versus building fine roots, leads to a 1 to 5% decrease in nitrogen uptake. So that's some, but it's not enough to get us there. The metabolic costs could be a lot bigger. So this is paying bacteria to fix versus paying mycorrhizae to help, or paying bacteria in the soil to break organic material down. So this gives us a range of 5 to 55%, which could be greater than 10% or could not. It says that we don't have a slam dunk of an answer here, but it seems reasonable. So, could there be selection against n-limited n-fixation? Yes, there could. One of the tricky things about this, though, is this is looking at the strategy of n-fixation. Not all, the plants that fix nitrogen can take nitrogen from the soil as well. So this doesn't tell us much about nitrogen fixers themselves. It tells us about nitrogen fixation as a strategy. The data I showed you in the beginning were for the abundance of nitrogen fixers themselves. So to understand why there are no nitrogen fixers, understanding why nitrogen fixation doesn't pay is part of the story, but it's not the whole story. We need to understand why it might be costly to be able to fix nitrogen, even if they're not fixing, or why they might be fixing even if they don't need it. And I'll talk about that in a bit more detail as we go on. So leading into that, an observation that I made while doing my PhD work in New Zealand has led down a path that I am still really excited about today, and that has to do with the ability of plants to turn fixation on and off. So I did my PhD work, the field work in New Zealand. This is the Franz Josef Glacier in New Zealand. Nice place to work, fairly picturesque. What you don't see in this is that this site gets about 6 meters of rain a year. So most of the time it looks like this. But I did actually get to see this at least on one day and take that picture, and now I can look at it. But most of the time I was huddling in the cold and being very wet. in that system, I was looking at fixation by plants as well as by bacteria living on their own and with a bunch of other things. Plants is what I'm going to be talking about today. So this is the plant system that I was interested in, Coriaria arborea. We looked at fixation by them, which means doing gas work and then digging a bunch of soil pits to see how abundant nodules are. And these are some of the data that I found there. So this was looking at 2 sites along the successional sequence, so a 7-year-old site and a 60-year-old site. These are the 2 youngest sites, and they're also the only 2 where end fixers exist. So that successional pattern I showed you in the beginning, generally end fixers, where they're there, drop out during succession, that holds here. The top panel shows nitrogenase activity, so that's the activity of each gram of nodule, how much nitrogen is fixing, and we measure it in a slightly different way. We can talk about that if anyone wants. The bottom panel is looking at the density of nodules, so this is how abundant are nodules across the landscape. And there's some variation. Red is summer, blue is winter. Some variation, but there is no trend. They fix exactly at the same rate, and they have exactly the same amount of nodules at both of these sites. If we were to plot this up per unit plant biomass or basal area, we get the same thing. This is really weird when you know that these 2 sites have the lowest nitrogen availability and the highest nitrogen availability of anywhere in this region of New Zealand, and in fact, in most temperate forests anywhere. That's a very low concentration of soil inorganic nitrogen, that's very high. If we plot this up with nitrogen mineralization rates or any other index of nitrogen availability, we get the same answer. These sites are at the opposite ends of the spectrum. Plants are fixing exactly the same amount at the lowest nitrogen and at the highest nitrogen ends of the spectrum, which means that they might be fixing nitrogen even when they have plenty in the soil, which seems kind of weird. When I went back to the States and talked to a lab mate of mine who was working in Panama at the time, he had been doing similar studies looking at some plants in Panama. So his data are in red here, mine are in black here. This is looking at nodule biomass, and this essentially tells a little story. In New Zealand, plants had exactly the same amount of nodule biomass and same fixation rate at very low and very high nitrogen concentrations. In Panama, at very low nitrogen concentrations, they seem to be fixing a fair bit on average, and at high nitrogen concentrations, they invariably turn fixation off, which that one made a lot more sense to me. But there's this contrast. Plants in New Zealand weren't turning fixation off, plants in Panama were. So I then went into the literature and tried to see what other people had found about this, and I could find some other evidence for similar dynamics for alder in the United States. So alder, red alder particularly in the Northwest, has been really well studied. It's used in forestry. And it often fixes a ton of nitrogen, even when there is a ton of nitrogen in the soil, which people had written. Yes, that's weird, but they do this. In the tropics, the plants that Alex was working with seem to turn fixation off as soil nitrogen goes up. Some acacia species in Hawaii seem to be doing the same, and other species that people have looked at since then in Panama and Costa Rica seem to be doing the same thing. So we see this apparent difference in nitrogen fixation strategy between higher latitude trees and lower latitude trees. One interesting thing to note is that these 2 trees are both actinorhizal fixers, and these are both rhizobial. So remember, in the beginning I talked about those 2 different types of nitrogen fixers, and those 2 types are not randomly distributed across the globe, they're clustered. This type tends to be distributed at higher latitudes, this type tends to be at lower latitudes, and I'll show you data on that later. So we started to see this difference in pattern. And what I wanted to do then was try to understand what are the consequences of those 2 different strategies, or particularly what are the consequences of obligate fixation for the nitrogen cycle and for the fitness of end-fixing plants in different environments. So we went back to the same model. We now modified it such that we're looking at nitrogen and phosphorus, and assuming that either one can be limiting or they can be co-limiting. And we wanted to specifically look at obligate fixation and try to understand what are the consequences of obligate fixation for fitness of these plants and for the nitrogen cycle in those areas. We specifically looked at the case of weak trade-offs. In fact, I think there are no trade-offs in the one I'm about to show you, to try to understand the consequences of obligate fixation itself absent these trade-offs. So obviously if we throw trade-offs in, fixation makes less sense. But if we look at a case where there are no trade-offs and fixers are obligate, then we can pinpoint obligate fixation as being the cause of whatever we're seeing. So these are simulations between obligate nitrogen fixers in the solid line and non-fixers in the hatched line. At the top, I'm showing you biomass of both types. At the bottom, I'm showing you losses of available nitrogen. So this is one of our indices of nitrogen availability. And on the horizontal axis, I'm showing you successional time. So I'm starting it like primary succession would start and just running the simulation to see what we have. For the first few years, N fixers do really well. So I set it up such that nitrogen is very strongly limiting to begin, and obligate fixers do very well. Fairly soon, they fix enough nitrogen to enrich the entire ecosystem. They overcome nitrogen limitation to themselves, and they also provide enough nitrogen such that all the other plants are not nitrogen limited. At that point, you see this big spike in available nitrogen, sort of like we saw in that second youngest site in New Zealand. And N fixers slowly are excluded from the system by non-fixers. And if we keep running this out, they stay at very low concentrate or at very low biomass indefinitely, whereas non-fixers go up and up and up, and eventually will flatten out. So this is exactly the pattern we see in temperate forests, and it all stems from simply the assumption that fixers are fixing even when they don't need to. One brief comment on obligate fixation. So when I say obligate, I don't mean that the plants need it to survive. I just mean that they're fixing at about the same rate everywhere we see them in nature. So it doesn't mean they have no control over it, it just means that they are fixing at that same rate. Alright, so what are the consequences of obligate fixation? Well, obligate fixers overcome N limitation and they fertilize their competitors. So if we understand why late successional N fixers have not evolved, it seems like we have to look for selective reasons, because phylogenetic constraints are not the answer. A trade-off between N fixation and N uptake, so some cost of N fixation, might be strong enough to select against the strategy of N